So yesterday, since we had done the operator and the repressor mutations, to summarize the repressor mutations, we are having these three conditions where I plus, I minus, where I plus is dominant to I minus, I plus, I is dominant to I plus, I minus D I plus, I minus D dominant to I plus. Now, what will be the scenario in presence and absence of lactose? If I just summarize the presence and absence of lactose, what will be the scenario? Then okay. So in this first case, absence of lactose, just a second. So if I write here, screen pointer option, then Lac Operon. Lac Operon is status. The status of Lac Operon. Here, absence of lactose. So let us take it this way, absence of lactose. In this situation, here we'll be getting a normal setup. The operon will be switched off. Okay, so the operon will be switched off. In this condition, where super repressor is dominant to the normal repressor and permanently almost binds to the operator, the operon will be switched off again, behaving like normal. And in case of I minus D I plus, as I explained yesterday, it doesn't bind, this protein does not bind to the operator, nor does it allow the normal repressor protein to bind to the operator. We will be getting a constitutive synthesis, that is the Lac Operon will be switched on. In presence of lactose, now if we give lactose to the system, so in presence of lactose, in case of normal, we'll be getting a plus sign, that is operon switched on. In case of super repressor, it is permanently switched off. And in case of I minus D also, it is permanently switched on. So keep it in mind. When we have this kind of mutation, the operon is permanently switched on. So the operon is under no more regulation. And in this kind of condition, as you are seeing, the operon is permanently switched off. So we studied these three repressor mutations yesterday along with the operator mutations. And if there is operator mutation also, the operon is permanently switched on for that DNA. Okay. So we studied the concept of cis-dominant gene. We studied the concept of trans-dominant gene. Now there are two more mutations that can be talked about. One of the mutations, there is no picture. I'll just explain it through this this particular, let me explain it to the operator one. Okay, so let's get into this one. So the one mutation is the promoter mutation. So if we have a promoter mutated, so remember, if there's a promoter mutation, RNA polymerase will fail to recognize the promoter. So in absence of lactose, no problem, the operon is switched off. But in presence of lactose also, the operon will be switched off because RNA polymerase does not recognize the promoter elements. So if we have a condition like this, P plus, that is promoter normal, P minus, that is promoter mutated. So keep it in mind that for this DNA strand, there will be no Lac production. But for this promoter element, there will be Lac production. So let us take this kind. It's very similar to the operator case. Point options, okay. See, suppose imagine this promoter is defective. This is the normal one, where we had that defective one. Okay, let's take this one. Suppose this promoter is defective. Let us imagine. So what will happen? In absence of lactose, repressor protein will bind here. And since this operator is normal, let us imagine this operator is normal, the repressor protein will also bind here. So overall, in absence of lactose, no net production of the enzymes. Imagine that this operator is normal. So what we are having a promoter mutation. So in absence of lactose, the operon will behave as normal and will have a no production status. The moment lactose is given in the system, what happens? Let's look in this scenario. So the lactose is given in the system and the inducer molecule will open up this repressor. The inducer molecule will open up this repressor. But as I said, we had a defective promoter. The RNA polymerase will recognize this promoter element, will sit here and start transcribing, but the RNA polymerase will fail to sit here. So there will be no production. So keep it in mind that the DNA on which the promoter is mutated, that DNA becomes defective, and that the promoter element is then exerting its effect on the structural genes present on that DNA. So promoter mutation is also a cis-dominant mutation. It behaves exactly same like operator. The difference is if operator is mutated, that DNA becomes permanently switched on. If promoter is mutated, that DNA becomes permanently switched off. This is the only difference. But the basic mode of action of the gene is same. Both of these elements, the promoter and the operator, are actually exerting their effect on the downstream structural genes, and both are cis-dominant mutations. Just a second. Finally, coming to the last category of mutations that are seen in the Lac Operon, that is the mutation of the structural genes. So if we look into the mutations of the structural genes, keep it in mind. Keep it in mind that during mutation of the structural gene, the gene which is mutated will be defective. For example, we are having in this situation Z minus, Y plus, A plus. So what is the status of Z? It is a defective gene, so we'll write a minus sign. Y plus is producing a normal protein, so we'll write a plus sign. And A plus is also producing a normal protein, so we'll write a plus sign, okay? In this case, if you look into the genetic setup of the lower strand, as you can see, the Z is normal, so we'll write plus. Y minus is defective, minus. A plus is again normal, so you're getting a plus. So it's very simple. If we have a defective protein, for that protein we use the symbol. If in presence of lactose, when we write the condition in presence of lactose, since Z minus is defective, we will write a minus symbol, because even though the protein is being produced, because the protein is defective in nature. Here minus does not indicate that the protein is not produced. Here minus sign indicates that the protein is produced, but the protein is not functional beta galactosidase. Hence the negative sign. If the protein product is normal, then we will be writing plus sign. So this is how the signs are involved. There's another kind of mutation known as nonsense mutation. I'm not getting into the details of nonsense mutation because that will confuse you. I'll just show you some examples of how problems are done. See, let us look into this particular picture. So we have studied the repressor mutations, 3 category. We have studied promoter mutations, only 1 category. Operator mutations, only 1 category, and ZYA mutations, only 1 category. Now, ZYA mutations usually occur when there is a wrong codon. Instead of methionine, serine has been inserted. So the codon has changed. So there is a change in one of the nucleotide base. Okay. Now let us look into a situation like this. See, what is the condition given? I plus, P plus, OC, Z minus, Y minus, A minus. So we write I plus, P plus, where is OC? Oh, this is written down. I plus, P plus OC Z minus Y minus A minus. Here we have the other one, we have I plus O P plus O plus Z plus Y plus A plus. So in your exam, problems like this will come. So they will be giving you a scenario like this. I plus P plus O plus Z plus Y plus A plus, right? And in the other hand, we have I plus P plus O plus I plus, no, this is not the I plus P plus OC Z minus Y minus A minus. Okay, so this is the problem. You have to say what is the condition of, now in your problem, you have to now state the condition of Z and Y in absence of lactose and presence of lactose. So you have to state this thing. So when you do this problem, just like as it is shown here, you just draw it as into two strands. For your simplified image, I'm showing it. I'm drawing it as two strands. I'm keeping it like this: I plus O plus P plus O plus Z plus Y plus A plus. This is one strand for me. And now I have another strand, I plus P plus, I'll draw it in a very simplified manner, OC Z minus Y minus A minus. So this is our scenario. So let us assume that in absence of lactose, what will happen? So there is no lactose in the system. We are having a normal repressor protein. So the repressor protein will come out. As you can see, the normal repressor protein will come out from this repressor gene and from this repressor gene. This repressor protein will go and bind to this operator molecule because this operator molecule is normal. But we are seeing an operator mutation here. So there will be no binding because OC, the repressor molecule, cannot bind. Since the repressor molecule cannot bind, so the polymerase will transcribe this mRNA. But as you see, we are having a defective Z. So no product functional beta galactosidase, a defective Y. So no functional beta galactosidase, a defective A. So no functional beta galactosidase. So in absence of lactose, we will write minus, minus. Now, lactose is given. So lactose has come into the system. So the moment lactose has entered the system, so what happens? This small inducer lactose or the allolactose binds with the repressor. Next moment, what do we see? Sorry, the repressor is removed. So let me. So the repressor protein was removed. Now we are having a functional operator. We are having a functional operator. RNA polymerase will sit at the promoter and will transcribe this mRNA. From this mRNA, we are getting a functional beta galactosidase, a functional permease, and a functional transacetylase. So in presence of lactose, we will get a picture like this. So this is how the problems are to be done. So this is one problem I showed you. I'll show you another problem. So let us do another problem in similar manner. Now let us choose this one. Let us choose, we did a OC problem. Let us do a repressor problem. So we have another problem, I plus, P plus, O plus, Z plus. This is how we write in our copy. Our problem is this one, Y plus. See, this is the problem I'm talking about. So I'm writing it in my copy. In your exam, a problem will come like this. So when I write in my copy, I make some distance so that I can imagine that there are two strands, I minus, P plus, O plus, Z plus, Y plus. Fine. So we have our problem now. Now let us make the table. We have to find the status of Z and Y. So Z, Y, Z, Y. Absence of lactose, presence of lactose. Now see, in absence of lactose, no lactose in the system, minus. So repressor protein, this is producing normal repressor. This has no product, but we all know that I plus is dominant to I minus. It is known to us. So this normal repressor will go and bind to this operator and also to this operator. So in absence of lactose, the operon is behaving as normal. The operon is switched off. So we will not get any Z or Y product. Now let us change the situation. Lactose is given. And what happens the moment lactose is given? The repressor will be removed from the operator region. So now polymerase sits on the promoter and transcribes the Z and Y. Here also transcription of Z and Y is occurring. So we are getting the product of Z and Y. We are getting the product of Z and Y. So in presence of lactose, we will be getting a scenario like this. So with this, we complete one part of the Lac Operon. Now the concept is that the Lac Operon, the concept what we studied till now was that of negative regulation. Because now we will be entering an area which is known as the positive regulation of Lac Operon. Now why negative? Let us assume this is the picture of the Lac Operon. I should come to this picture and let us look at this particular slide. In absence of lactose, how does the normal operon behaves? In absence of lactose, we have seen that the repressor gene produces the repressor protein, and this repressor protein can go and bind to the operator region. The binding of the repressor molecules to the operator DNA results in no production of the mRNA for Z, Y, and A. So there is no mRNA production because as the repressor remains bound to the operator, RNA polymerase cannot bind to the promoter, and the main thing is that there is no mRNA production. Hence, this is known as negative control. Negative or positive control depends on the fact that whether the controlling elements are allowing the gene to be transcribed or not. Here, the repressor controlling element is not allowing the gene to be transcribed. Hence, the normal condition via which repressor exerts its effect is a negative control. As I said, there is no mRNA production. There is another phenomena known as Catabolite repression or the glucose effect, which is known as the positive control of Lac Operon. So now let's move on to the positive control of Lac Operon. Okay, now I think so. I told this in my yesterday's class that the operon system ensures that Lac operon will be exposed at expressed. The Lac operon will be expressed at high levels only if lactose is the sole carbon and there is no glucose present. The preferred carbon sugar preferred by E. coli is glucose. So if there is glucose present in the system, then glucose is always the preferred carbon source because it can be used directly by the glycolytic pathway to produce energy. Or whereas in case of lactose, it has to be converted to glucose first, which a step which needs energy, and therefore more energy can be obtained for the cell from glucose than from other sugars. So when there is a mixture of glucose and lactose present in the system, E. coli ensures that all the glucose is exhausted first and then only lactose will be utilized. Now the question that automatically arises in our mind is that we have seen the Lac operon. So the Lac operon has a I plus, P plus, O plus, Z plus, Y plus, and A plus. Okay, so this is a known operon, a known Lac operon. Now, in the system, there is lactose. So if there is lactose, in the system, we have both lactose and glucose, right? So if there is already lactose present in the system, then this lactose will remove the repressor from the operator. There is lactose in the system. So this lactose will obviously be converted to allolactose and will remove the repressor from the operator. And then operon and the polymerase will not have any problem in transcribing this downstream structural gene. Then how come, in spite of lactose and glucose is present, lactose is not utilized, but glucose is utilized? So this phenomena of utilization of glucose is known as, this phenomenon is known as Catabolic Repression and is done by a protein known as the Catabolite Activator protein, which pairs with the cyclic adenosine monophosphate. So I have come to show you this picture to relate one thing. Remember, glucose, when glucose is present in a cell in the form of glucose, then if there is very high glucose in a cell, we have to assume that there is very low ATP in that cell. This is inversely proportional because we know that in the glycolytic pathway, glucose is broken down to produce an ATP. Further, the product of the glycolytic pathway that is then converted, then it's passed on through the mitochondrial Krebs cycle to produce further molecules of ATP. So if glucose as a whole is present in the cell, that means we have to assume that the glycolysis and the Krebs cycle are not operative. So no ATP production. So if high amount of glucose, no ATP. But if low amount of glucose, that means glucose has entered the glycolytic pathway. The moment the glucose has entered the glycolytic pathway, that is, the breakdown of glucose has started, which indicates that ATP production has increased. So we have to keep this in mind. So this is an inverse ratio. So if there is more amount of glucose in the cell, low ATP, low amount of glucose in the cell, high ATP. How do we relate now? How do we relate ATP to catabolite repression? Let us look at the structure now. ATP in the cell is broken down to produce via adenylase cyclase to produce CAMP, cyclic adenosine monophosphate. So I can now logically say that if there is high glucose, low ATP, low cyclic adenosine monophosphate. If there is low glucose, high ATP, high adenosine, cyclic adenosine monophosphate. So we can draw a relationship here. So I can write here, indirectly I can write here that if there is low glucose, GL, I mean, there is high ATP, which indicates again that we have high cyclic adenosine monophosphate, right? So this molecule is now important. This molecule is only present in the cell when their glucose amount has lowered. So this is important. If there is high glucose, low cyclic adenosine monophosphate. If there is low glucose, then there is high cyclic adenosine monophosphate. So now let us look into this story. Now in the cell, we are having high amount of cyclic adenosine monophosphate. As I related, that high amount of cyclic adenosine monophosphate indicates that glucose has now become very low in the cell. This pairs with a protein known as Catabolite activator protein. This is present in the cytoplasm, but in its inactive form. It can become activated only when cyclic adenosine monophosphate is added to it. So cyclic adenosine monophosphate binds with the catabolite activator protein to form the CAP-CMP complex. This is a transcription factor. So you know that there are these factors, transcription factors, that helps in enhancing the transcription rate. This transcription factor recognizes an entry site in the promoter element of Lac Operon, which is known as the CAP site. This is a site present in the promoter region of Lac Operon. So the moment we have an active CAP-CAM complex, which is possible only when glucose has almost been exhausted, the CAP-CAM complex then binds at the CAP site. The binding of the CAP-CAM complex now facilitates the promoter to enter the entry site, facilitates the polymerase. It facilitates the polymerase to bind to the entry site of the promoter element of Lac Operon and start transcribing the mRNA. In absence of this CAP-CAM complex, the polymerase can come and bind to the promoter element, but the binding is not that efficient. And hence, even though in absence of lactose and glucose, there is no repressor molecule present in the operator. And if the polymerase wants, it can bind to the promoter, but the polymerase binding is not efficient. Hence, it cannot efficiently bind to the promoter, and hence it cannot transcribe properly. Only when the glucose has been exhausted, we get this CAP-CAM complex, which acts as a transcriptional activator. It has a site in the promoter region of the Lac Operon. It binds on the CAP site and facilitates the entry of the polymerase into the its entry site, and thereby helps the polymerase to increase the rate of transcription. Why is this called a positive control? Because the binding of this molecule to the promoter element is actually resulting in the increase in mRNA production. So mRNA production has become high. Hence, it is known as positive control of Lac Operon, as compared to the repressor molecule, which when binds to the operator results in no production of mRNA. So this is the difference. So this is known as a positive control because mRNA production has increased, and that is known as the negative control because repressor protein results in switching off of the operon. So this is catabolite repression. Pointer options, sorry. So you see, this is the E. coli cell. It has glucose and lactose. Both are entering, but you see it is utilizing glucose first. And then only lactose is used. Lactose is a double sugar, glucose is a single sugar. That is why they have showed it in that manner. So this preferential utilization of glucose over lactose is done, is controlled by this catabolite repression. So when we have, this is important, listen, this is what I was saying. When we have both lactose and glucose in the system, you see the slow rate of transcription. There is no repressor molecule bound to the operator because lactose is already present in the cell. In spite of that, very little RNA is made. How and why? That was a question. And only when glucose is totally exhausted, you will see, glucose is totally exhausted, you will see that the lactose will be utilized. So once the glucose is totally utilized, then lactose metabolism starts. So transcription increases when glucose is gone and rapid lactose metabolism starts. Now, just now I showed you the structure of cyclic adenosine monophosphate. This is the breakdown product of ATP. ATP is broken down to produce cyclic adenosine monophosphate by the enzyme adenylase cyclase. It's a very vital enzyme, which is further broken down to form adenosine monophosphate. So this was the relationship I was talking about. If there is high glucose, low ATP, low cyclic adenosine monophosphate. Low glucose, high cyclic adenosine monophosphate. This is relation. We have to keep it in mind. So lactose present, glucose absent. So we are now having cyclic adenosine monophosphate. See, active CAP-CAM complex. There is a LAC promoter, the CAP-CAM complex. So we have this LAC promoter, just once more, let's see. We have this LAC promoter, and in the LAC promoter, the CAP-CAM complex has come and bound. Once this has been bound to the CAP site, what happens? This is the entry site of RNA polymerase, and this CAP-CAM complex binding will now facilitate the RNA polymerase to enter into the entry site. So you see, it has facilitated the RNA polymerase to enter, and we are getting enhanced transcription. So this was the story of, so glucose and lactose abundant, low cyclic adenosine monophosphate. First, glucose is utilized. See what happens. This is important. So when there is glucose and lactose together, unless and until glucose is exhausted and cyclic adenosine monophosphate is produced, we do not get an enhanced rate of transcription. You see what happens here, the polymerase comes and sits, but the rate of transcription is low. So it's here, slow transcription efficiency, because the polymerase is not guided by the CAP-CAM complex. Transcription occurs, but at a very low rate. Only when glucose is totally metabolized or exhausted, we get high cyclic adenosine monophosphate, which binds to the CAP to form the active transcriptional activator, and then the RNA polymerase transcription rate increases. So let's go over that once more. Yeah. So slow transcription efficiency. See, once glucose is exhausted, then only cyclic adenosine monophosphate forms. So it is the amount of glucose present in the cell which actually at times determine how fast the Lac Operon will operate. How fast the Lac Operon, how fast will the transcription of the Lac Operon take place. So the catabolite activator protein and the CAP-CAM complex is a transcriptional activator. Why is it called positive control? Because binding of the CAP-CAM complex enhances the transcription of the mRNA. While compared to the repressor molecule, if you look at it, if you look at the repressor molecule, you can see that the repressor molecule binds to the operator, and as a result of which no mRNA production is occurring. Hence, it is called negative control. So the repressor inhibits transcription, the CAP-CAM enhances transcription. Since the CAP-CAM enhances transcription, it is positive control. Since the repressor inhibits transcription, it is a negative control.